Well, good evening, everybody. Thank you for coming out on the evening of a 3-day weekend. So, this is the first time I've tried one of these. Actually, I tried one of these maybe about 15 years ago, so I'm happy to try it again. So this is not a review session, as I said in the email that I sent out. This is a sort of a let's look at the stuff in a different kind of a way. And so I'm open to a variety of things. I'm open to questions. I do have something I've put together to present that will sort of provide the basic framework of what I'm going to do. And I'll tell you a little bit about my thinking with this. So I'm still wrestling with the nature of the classroom that we're in. I am used to a little bit more interaction than what I'm having. I'm not complaining, so don't mistake that. So I want to have that. I find that I like to talk, and you guys know I like to talk, but I find that if I can make that more interactive to you, that's better. And in the absence of that, I'm thinking that maybe there's something I can do to help sort of get through the material. And the sort of impetus for me was the blood clotting lecture. When I gave the blood clotting lecture, I think I've told several of you, I didn't like that lecture. I just wasn't happy with it, and I felt that it was just way too much information for what you needed to learn about it, and I got to thinking, well, maybe I could format that a little bit better and make that a little bit more understandable to everybody. And so that was the starting point, and then I sat down and I said, well, why don't I just take everything since the last exam, which is what everybody's interested in anyway, and reformat it. So I'm not going to change the PowerPoints or any of that sort of stuff. But what I decided to do was go back and make outlines for everything, because I've had pretty good feedback people have given me on the fact they like the outlines and so forth. And as I've said before, I've usually lectured from outlines, which I like a lot. So I went back and made outlines, starting with the very beginning of the material since the last exam, and taking it then all the way up to the lectures I haven't given yet. So for example, I stopped when I got to glycolysis. Okay, so glycolysis lecture I'll give on Monday, and probably we'll have the material for the exam that will go up through Monday. That's my tentative plan. Okay, so we probably won't get all the way through glycolysis. If we do, great. If we don't, that's great. That's fine. So probably Monday is where I'm going to stop the material for the exam. So I haven't put that glycolysis stuff on here yet. So what I thought I would do, if it's okay with you guys, would be I would just sort of start and say, what if I were a student taking this? All right? How would I be looking at the material? And since the flip side of that is I'm the guy that's writing the exam, what do I think that students should be looking at? So I kind of put the two of those together in what I've written and what I'm going to talk about. Does that make sense? I'm not going to give you the questions. Okay? So if you came here looking for the questions, I'm not going to give you the questions. But I do have things that I think I can help you to focus on and understand, because there's a lot of material. There's an awful lot of material that's there. And you look at the length of just the outline that I've put together, and you can see there's a lot of material there, right? So I don't need to tell you that. So does that seem reasonable? Okay. So as I'm going through things, you want me to say something a little bit more about anything, then just blurt it out, whatever, and I will do that. I have sort of taken this, and what I did in making this outline was I didn't look at the keynote slides. So there's probably some slides there that just aren't going to be reflected in the outline, but I did look at all of my highlights in writing this, which I think are a reasonable representation of what's there. So this came from the highlights, and as I think I've said in the class, when I write an exam, I use the highlights to write the exam. So this seemed like a good way to just kind of capture all that. I will make this document available, so you don't need to write it down, okay? So I'll put it on the class webpage, and you can have that, okay? I see everybody writing down furiously, right? So I think the worst way to study is to try to write as fast as the professor talks, right? And try to copy something down, and I'm trying to get, and then all of a sudden the slide changes, and then you're lost, right? So I think that's really a dumb way to teach. So I try to avoid that as much as I can. All right. Well, why don't we go back to rewind the thing, go back to the beginning of the new material since the last exam. So we started this material with, with catalytic strategies. So if I look at this from the top down, okay, what's there is I have catalytic strategies, and you can see the biggest type and to the left are the major points that I've talked about. We had a couple lectures on catalytic strategies. We had a couple lectures on allostery and regulation. We had a couple lectures on carbohydrate structure. We had a couple lectures on, and what does that say, receptors? That should be signaling. I guess I didn't know that set up right. So G-protein coupled receptors, which were involved in signaling. I should have had signaling as a category. I talked about receptor tyrosine kinases, RTKs. I talked about steroid hormone signaling and metabolic energy. Those are the main points of what I talked about in class so far. Okay? So those signaling things all should have been one big group instead of individual groups there. So in terms of big categories, there's only about 5 or 6 big categories that we've had since the last exam. Okay? So looking at that back down, let's now break those down into the things that I have talked about. All right. So talking first about, let's see, I should get my laser pointer and try to laser point a little bit. Okay. Okay. So, there we go. All right, so talking about catalytic strategies. When we think about catalytic strategies, how do enzymes do what they do? That's really what's up, right? How do enzymes do what they do? We saw several things in the catalytic strategies, but I tried to sort of bring them all together. Every catalytic strategy that I talked to you about involved nucleophiles. Remember that, okay? Every catalytic strategy we talked about involved nucleophiles. And we talked about how those nucleophiles were made in several ways. The first one that I talked about were the serine proteases. So the serine proteases were nucleophiles. And by the way, you see I'm not going to show you figures here for the most part. If you want, I can pull up figures for something, but I'm just going to kind of lead you through this verbally. Okay? That's my plan. All right. So the serine proteases were the ones that had the catalytic triad. The catalytic triad, of course, aspartic acid, histidine, and serine, right? Remember that the serine proteases are a group of proteases. What's a protease? Cleaves a protein, right? Breaks peptide bonds and cleaves a protein, okay? So a serine protease is called a serine protease because it has a serine that plays a critical role in that catalytic process. And you know that that serine is part of the catalytic triad of a serine protease. So now those terms all should link together. Okay? So there's the catalytic triad. Briefly, I'm not going to take you through mechanism here, but briefly, that catalytic triad works by the binding of the proper substrate. Where does the proper substrate bind? In a serine protease. Anybody remember? S1 pocket, right? So the S1 pocket determines the specificity. The proper substrate binds in there, you get that shape change. When that shape changes, the catalytic triad, the geometry of it changes. Okay? The geometry changes. The aspartic acid moves closer to that histidine ring. The electrons in that histidine ring shift over closer to the proton on the serine. And that proton on the serine, which is positively charged, says, oop, I jump. And it jumps over to that histidine ring. That leaves behind an alkoxide ion. And that alkoxide ion, we've made our nucleophile. We've got a nucleophile. Okay? So all these different proteases that I talk about, they're all making a nucleophile. And in every case, that nucleophile is attacking the carbonyl carbon of the peptide bond. Okay? The carbonyl carbon of the peptide bond. People say, what's the carbonyl carbon? Well, the carbonyl carbon. That is the carbonyl carbon. Okay? So it's attacking that bond. When it attacks that bond, we see some electronic change that happens. As I say, I'm not going to go through the mechanism, and I don't expect you're going to go through that mechanism. You're not going to push electrons on this exam. All right? This bond becomes unstable because of the attack, and it falls apart. That's where the oxyanion hole comes in. It helps to stabilize the intermediate so it doesn't attack the enzyme, and then it falls apart. Yeah? Yeah, what's the hole? What's the oxyanion hole? So the oxyanion hole is literally that. It's a little pocket in the active site. It's just a little pocket. And it's a fairly hydrophobic pocket. You don't need to know that. Okay? It's a fairly hydrophobic pocket where this intermediate just sits. There's nothing for it to react with. It's there. It's there. It's right there. So you got to understand, this whole active site thing is just a little cave, and this is a little pocket in that cave is really all it is. Okay? Yeah?
So would it be safe to say that the oxyanion hole, like, it's just there, but only when it's in
No, the oxyanion hole is there. Okay? So just like you would have a chamber of you'd have a cave, and you might have some little side room. It's just sitting there the whole time. Okay? So there's just a place for this guy to sit, and there's nothing for it to react with. This thing is fairly reactive when it's been attacked. And you don't want this thing to just randomly react with the enzyme. Because if it did, you would create a suicide inhibitor. It would react once, and that would be the end of it. So the oxyanion hole is there to keep this guy from reacting with the enzyme so that this bond can fall apart instead of binding to the enzyme. Okay? Now you want this guy to bind to the enzyme, but you want it to bind where? You want it to bind where that hydrogen was on that alkoxide ion. That's the specific place you want it to bind. Okay. Now, if you'd like, I will just talk you through the steps of that. And if you'd prefer not, I won't. Will that confuse you if I go through that now or not? No? Okay, everybody's saying no. Okay. If it does, then I won't. All right. So let's talk about the steps in that process. We've done the first part. We've done the binding. Second part, the formation of the alkoxide ion. Third part, nucleophilic attack. I'll slow down. Fourth part, this guy is sitting in the oxyanion hole and the bond falls apart. So this bond right here is going bye-bye. Bond falls apart. At this point, we've gone through the fast part of the process. We've broken the first peptide. We've broken the peptide bond. One part of the peptide goes flying away because there's nothing holding on to it. Okay? Now we start the slow process. When this bond breaks, this guy ends up getting bonded to gets bonded to the alkoxide ion. That's what happens. Okay? It's bonded to the alkoxide ion. It's a covalent bond. Got to break that bond. That bond gets broken when water comes back in. So this is the slow step. Step 5, entry of water. Now, yes, ma'am. Which bonds? So this bond is broken. This guy is out of the picture now. Okay? This guy is now bonded to that alkoxide ion because this made the attack on that carbonyl carbon. Okay? Right. And actually, I've drawn this. Hold on here. Where's my thing? I've got 4 bonds. I should have 5 bonds. Sorry. Okay? So it's bonded. All right? Now, we've got to release it. Water comes in. That's the 5th step. And by the way, you know, what we call a step can vary. Was sitting there in the alkoxide in the oxyanion hole a step, or was the falling apart of the bond a step? You decide. All right. So I'm not going to say, well, what's the 5th step in this process, because that would just be dumb, right? But if you know the process, then you could if you give it 12 steps or 13 steps or 20 steps, I don't care as long as you understand the steps, right? Okay. Water comes in. Water has got to become a nucleophile. How does water become a nucleophile? Well, you got that histidine sitting there, and that histidine's full of electrons, and water's got a proton, and exactly the same thing happened with the serine hydroxyl happens to water. Water loses a proton. Now you've got a reactive hydroxide. It's a nucleophile. Guess what it's going to do? It attacks this thing. I don't give it a name. Call it Larry. Okay? I do that especially with my BB350 class. We'll get to something and it's got this long, convoluted name. And I say, why don't we just call it Larry? Okay? And the class really likes that because they can remember Larry, but they don't remember the long, convoluted name. And if the long, convoluted name doesn't matter, what difference does it make? I tell you what. You can put Larry on the exam because, A, I'm going to tell you, I'm not going to ask you for the name of that. But if you put it on there, the TAs are all going to come to me going, why the hell are people talking about Larry? Because that happens every term, right? Who's Larry? Do that. I'll love it, and they'll be all puzzled, right? And then Larry has to be released, right? So, okay. So Larry can get released. Larry's going to get released, guys. All right? That happens because you've got this hydroxide that's been activated. It's a nucleophile. It goes. It attacks this thing. Bang. Bond. It attacks. And now everything goes flying away. This guy gets released. Okay? This guy gets released. The hydroxide on the side chain of serine gets restored, and everybody's done. Make sense? Yes? Yeah. So what binds to an S1 pocket? It depends on the serine protease. So different serine proteases have different S1 pockets. All right? So in the case of trypsin, trypsin is a protease that cuts adjacent to lysines and arginines. All right? So at the bottom of the S1 pocket is a negatively charged molecule because the side chains of lysines and arginines are positively charged and they're attracted to it. It's the R groups. That's correct. Okay? In the case of chymotrypsin, it has a fairly large nonpolar pocket, and that's why it has things like the phenylalanine side chain that goes in there or the tryptophan side chain that goes in there. Make sense? Okay. So that's what's happening in the S1 pocket. All right. All right. So that's what's happening in the mechanism. Now, the other thing I talked about with respect to serine protease is actually related to human health. And with human health, I talked about serpins. And serpins are just simply a category of protease inhibitors. The name serpin means serine protease inhibitor. That's what serpin stands for. And one of the serpins is alpha-1 antitrypsin. Alpha-1 antitrypsin inhibits a couple of enzymes, one of them being trypsin, which is a serine protease, the other being elastase. And elastase is the critical one because elastase is in our lungs. What's that? And so if it's not properly controlled, elastase gets out of control and causes emphysema. Right? Okay. So how does it get out of control? Well, if the alpha-1 antitrypsin gets somehow damaged so that it can't function, then instead of controlling elastase properly, what it's doing is elastase just goes crazy. Elastase starts attacking lung tissue, and that's what causes emphysema. How do you get damage to your alpha-1 antitrypsin? Oxidative damage. Oxidative damage happens from reactive oxygen species. The best way to get that is from smoking. There's a methionine side chain that gets damaged by a reactive oxygen species. When that happens, it can no longer bind to elastase, and that's why smoking is linked to emphysema. Okay? Okay, I'm getting in more detail here than I was going to do, but that's fine. Does that help? Okay. All right, other proteases. The other proteases differ a little bit in their mechanism, but every single one of them is making a nucleophile. A cysteine protease, instead of having a catalytic triad, it's got a catalytic dyad. I didn't call it that, but that's what it is. The dyad consists of histidine and a sulfhydryl of cysteine. So instead of pulling a hydrogen off of a hydroxyl, a cysteine protease is pulling a hydrogen off of a sulfhydryl, leaving a sulfur anion. That is a nucleophile, and it goes through exactly the same thing we saw the serine protease do. It attacks that carbonyl carbon. We see the bond fall apart. We see water come in to release it, and everything's done. Okay? Exactly the same things happen. They happen in a serine protease, except that instead of having a hydroxyl of serine, we've got the sulfhydryl of cysteine. Okay, metalloproteases. Metalloproteases have a metal ion that's holding on to a water, and that proton's got to get pulled off of water, which becomes the nucleophile. Exactly the same thing happens. It attacks the carbonyl carbon. The bond breaks. But the difference here is that the thing that's doing the attacking is the hydroxyl that isn't attached to anything. It's not attached to the protein. So there's no covalent bond between the peptide and the enzyme. I'm sorry, the peptide and the molecule being cut. So there's no need for a second step. You have one step. Cut. Both of them go flying out at once. Okay. Aspartyl proteases. Okay. Aspartyl proteases use aspartic acid, 2 aspartic acids that go out and do exactly the same thing that we're talking about. And they activate a water. Activate a water. Water attacks carbonyl carbon. Carbonyl carbon breaks the bond with the amine, and everything flies away. So, like the metalloproteases, the aspartyl proteases don't have a covalent intermediate with the enzyme, so it's a one-step and out of there reaction. Okay. Carbonic anhydrase. Carbonic anhydrase. I went through the steps with that. And one of the things about the carbonic anhydrase was that what carbonic anhydrase is catalyzing is, anybody remember what it's catalyzing? The formation of carbonic acid from water and carbon dioxide. So it's basically solubilizing carbon dioxide. To do that, we've got to have a nucleophilic attack. The nucleophilic attack is done with water, like we saw with the other things we've talked about. And here, water has to basically ionize by itself. It's a little bit different than what I said with the aspartyl proteases and the metalloproteases. I didn't go through that in class. But water is ionizing. And the reason we see this enzyme having greater activity as we get a higher pH is it facilitates that ionization of water. So if this was the enzyme that I showed you, if you plot the pH versus the turnover number, and the turnover number went up to like 1,000,000 per second at pH 9, and it was about 1,000 down at pH 5. The reason is because water will ionize more easily at pH 9, and that ionizing of water, that is the loss of the proton, is critical for the action of carbonic anhydrase. I'm going to slow down. Is this helping? Is this making sense? Okay, don't say yes if it's not. I'll never forgive you. Okay. All right. Now, those were the mechanisms that I talked about for those enzymes. The next big lecture that I gave had to do with how enzymes were controlled. There were 2 big things I talked about there, allosterism and covalent modification. I also mentioned in class a third mechanism. Does anybody remember the third mechanism? It was the production of the enzyme. Very good. I would give you extra credit, except for I didn't give a chance for everybody to do it, so I can't give you extra credit. But I have decided that's one way I can get everybody to participate, is I give extra credit in class. Anyway, so the synthesis of the enzyme is the third mechanism. I am right here, allosteric regulation. You look astonished. Is that? Oh, so I'm talking about how enzymes are controlled, how we're regulating enzymes. So the first part of what I said was 2 of these. One is allosteric, second is covalent. The third mechanism then is whether or not the enzyme is made. I didn't talk about that in class because we haven't had a chance to talk about gene expression. And I will talk about that next term. But I want you to be aware of the fact that there are 3 ways of controlling an enzyme. Okay, allostery. The best example I gave you was ATCase. A couple points with ATCase. Now, maybe I should add this to my, I'll do this right here. I'll add this to my outline. Because there was a very important point I made about ATCase I didn't put on here, and that was this. Right. So why is feedback inhibition important?
It's kind of an interesting phenomenon when the end products end up shutting down things.
So the end product shuts down the whole thing, and how does it do that? That's fine. Okay. Anybody want to say anything? Yeah. Binds to it, so it's an allosteric interaction. And the critical thing here is the last molecule is inhibiting the first enzyme. You stop a whole pathway by stopping the first enzyme. That's the really critical part of feedback inhibition. Excuse me. You knock out the first enzyme in the cycle. It doesn't matter about controlling all the other ones. And it's for that reason that cells don't control every enzyme in the cell. Because you stop the whole pathway by stopping the first one. There's no need to have controls on the other ones. So feedback inhibition is a very, very efficient way of controlling an enzyme. Okay. Now, feedback inhibition almost always, in fact, feedback inhibition always uses allosterism. One of the problem questions I gave you said, why don't zymogens use feedback inhibition? Maybe you want to take a stab on that one? Why don't zymogens exhibit feedback inhibition? They're not active, and what does it take to make them active? It takes covalent modification, right? And so when we're talking about feedback inhibition, we're talking about binding something, because something is feeding back and binding to it. The binding doesn't activate a zymogen, because that takes some kind of a covalent modification. Right? So zymogens are always activated by covalent modification. Always activated by covalent modification. All right. I haven't finished. I should finish allosterism before I talk about that. But is your question? I'll let somebody say something. Okay. All right. ATCase. Very interesting enzyme. It's got regulatory subunits. It's got catalytic subunits, and they're all held together. There are 6 of each. 6 regulatory subunits, 6 catalytic subunits. The regulatory subunits bind 2 molecules. Binding of those 2 molecules affects the enzyme. One of the molecules that the regulatory subunit combine is ATP. ATP is an activator of the enzyme, which I said when we talk about allosterism, we talk about turning the volume up, and that's converting the enzyme into the R state. The R state is the more active state of an enzyme. ATP turns it on because ATP is an indication, hey, we got plenty of energy. Let's go make whoopee. Right? That's what ATP is telling the enzyme. Making whoopee means the cell's going to divide, guys. And this enzyme is critical for that decision because this enzyme is making the nucleotides that's going to be necessary for DNA replication and cell division. Energy is an indicator the cell is ready for that. If there's plenty of ATP, let's activate the enzyme. CTP, another nucleotide, binds to the regulatory subunits, turns the enzyme down or off, depending on how you want to say it. And I'll take either. But remember that it's a volume change. It's turning it down. It's converting the enzyme into the T state. The T state is active. It's just not as active as the R state. That's CTP. CTP, sorry. I may not have said it. I get blathering and I forget to tell you what it is. CTP is, of course, a pyrimidine. And this enzyme is catalyzing the first step in the reaction of pyrimidines. So if we have plenty of CTP, we don't want to make more pyrimidines. So this is the feedback inhibition that turns it off. Question? CTP will never turn in the R state because CTP is an indication we got too many pyrimidines. You don't want to turn in the R state if you got too many pyrimidines. We'll talk about this next term, but imbalances in amounts of nucleotides, if you have too much of one versus another, you favor mutation enormously. So cells put a lot of control in making sure they don't make too much of any one nucleotide. Question?
Yeah, I'm a little confused. So CTP is the end product of.
The pathway.
Okay, but that's pyrimidine biosynthesis, right?
That's correct.
Do you want us to know? But then it inhibits.
It inhibits the first enzyme in the pathway, which is ATCase.
So it's just like a cycle.
It is a cycle. So if I look at what's happening, let's see. Hold on here. Maybe somebody could give me a piece of paper. I'll draw it out and show you what I'm talking about. Okay. So one of the common mistakes that students make, this should be here, right? .cam. There, no, here it comes. Okay, there we go. All right, so one of the common mistakes students make is this. Here's ATCase. Okay? If we look at the synthesis of pyrimidines, we see ATCase catalyzes a reaction here. We see another reaction, we see another reaction, we see there's actually about 10 reactions. I'm only going to draw 4 here. The mistake students make is they want to make CTP be the product right here of the first reaction. It's not. CTP is the end product. And they confuse the interaction of CTP with ATCase for ATCase catalyzing the production of CTP. It doesn't. This enzyme down here, which I didn't even put a name on, it's called CTP synthase, is actually what catalyzes the synthesis of CTP. So don't confuse ATCase's activity, which is making a precursor, with the end product, which is CTP. Make sense? Okay. All right. And we'll go back to laptop. Oop. Laptop. Here we go. Okay. So that's the ATCase story. Now, well, I'm sorry, one more thing. Yeah. Because that's the way it evolved. That's always an unsatisfying answer. Why is aspartate the only one that does that? Because it's using 2 substrates to make the next thing. Why is aspartate the only one? Aspartate was the only one it needed, so it evolved that way. Yep. But aspartate's the 3rd allosteric effector. And instead of binding to the regulatory subunits, aspartate is a substrate. Now here is where aspartate comes in. It is a substrate of this and makes a molecule here, XYZ we'll call it. Okay. Aspartate is a substrate of the enzyme. That's different from CTP or ATP or any of that. Okay. So aspartate is binding to the catalytic site, not to the regulatory site. There we go. Okay. Okay. Does that answer? Okay. Okay. All right. Let's move on. But I'm going to take a long time at the rate I'm going, but that's fine. Covalent modification. So here was what got me started. I didn't like the way I talked about that. So let's talk a little bit about covalent modification, and then this will lead us into blood clotting. All right. So I talked about zymogens. Zymogens are inactive forms of enzymes. They're inactive. They have to be covalently modified to become active, and usually, not always, but usually that modification involves a cleaving of a peptide bond. Cleaving a peptide bond opens up and exposes the active site that wasn't exposed before. Okay? There's a variety of covalent modifications that can happen. I'm not expecting you're going to memorize all those. I just show them to you so that you know that there's a bunch of them. All right? You can put a phosphate on. You can put an acetyl group on. You can put a formyl group on. You can put an acyl group on. All these various things that you can put onto a protein. These get covalently modified in that way. Okay. Now, zymogens are very important for controlling proteases because proteases you don't want attacking the tissue that makes them. And that tissue that makes your digestive proteases, for example, is your pancreas. You don't want protease is attacking the pancreas because that's how you get pancreatitis. All right? So these guys are made as zymogens. They travel further into the digestive system where they get activated. And I showed how you had the trypsin that played a major role in that activation, activating a lot of those proteases. And a question back here. How is it they're not made when they're synthesized? Okay. So it's a good question, actually. How are they not activated at the place where they're synthesized? And that's because there's no active trypsin at that point, hopefully. Because if there's active trypsin there, they're going to get activated, and they are going to cause the pancreas to be damaged. That's how you get pancreatitis. So you have to have some sequestration to keep that active trypsin from being at that point. Okay? And that's actually, the answer to that is actually an anatomical one that we're not going to go into here. But suffice it to say that that thing is set up so that you don't have that happen. If you do have it happen, you get pancreatitis, and that can be fatal. It's a serious thing if you get that happening to you. Okay. Blood clotting. Let's look at blood clotting in a little different way than I talked about it in class. So I spent some time thinking about how I wanted to simplify blood clotting for you and go back a little bit to the way I've taught it before. All right? Blood clotting happens, all right, through a very complicated set of cascading reactions. Okay? Now, there are two major components that start it. A cellular response followed by a molecular response. Okay? So what starts everything off is damage. You get damage to a blood vessel. Okay? That exposes some collagen that's not otherwise exposed. And now the platelets kick in and say, whoa, we got a problem. The platelets are this part of the cellular response. They're the first responders. They go to that site and they start binding. Okay? They go to that site and they start binding. Now, there's a variety of things that happen there, and I've mentioned some of them here. There's a release of calcium. There's the activation of the von Willebrand factor, and there's the release of thromboxane. Anybody remember why thromboxane is important? Makes them sticky. So all these things combine together to begin to put a plug where that hole is. It's not the clot, but it's the beginning of the process where that clot will form. Now, the calcium from the cellular response is what, and part of the activity of the von Willebrand factor, these two guys right here play roles in the molecular response. That's the second part of the process. So we have a cellular response, we have a molecular response. Everybody with me so far? Now, the molecular response gets broken into two components. And Megan, question. Okay. Okay. So what did calcium and the von Willebrand factor do in the molecular response? Calcium is part of that first phase of activation of the molecular response. Okay, so the calcium is one that plays a role in both. So the calcium is the one that's released here and helps activate things down here. Now I'm sparing you the details about how calcium is doing that down here. And what I'm talking about here with respect to blood clotting is what I'm going to expect you're going to know about blood clotting. I'm relieving you of a lot of the detail of what I talked about in class about blood clotting. If you want to know more, I'd be happy to sit and talk with you more. But I'm not going to go through with people here because I don't want to get people going, oh my God, here he goes again. From where is the calcium released? So blood cells, all cells have calcium stores within them. So they're just releasing from what they've got inside them. Mm-hmm. Yep. And that's a part of the cellular response. That's correct. You need the cellular response to activate the molecular response. That's right. You are right. Okay? And you can see what I've got down here is what I'm going to talk about. So you don't really see them popping up in here, but you see, for example, that actually, I didn't even put it on here, but calcium is responsible for activation of something called factor VIII. And I'm not holding you responsible for that. But that's part of this cascade that happens down here. Question? Calcium is an ion, so yeah. Calcium is a very small molecule. Von Willebrand factor is a protein. Von Willebrand factor is also involved with factor VIII, which I'm not holding you responsible for, okay? So both of those are playing roles relative to that. The factor VIII plays a role in the activation of factor 10. So I'm starting with factor 10. I'm not going through all that other stuff to hold you responsible. Okay? The reason I want to talk about factor 10 is because factor 10 is the place where the two molecular pathways converge. The two molecular pathways are the extrinsic response and the intrinsic response. They converge on making factor 10 active. Excuse me. Make sense? I'm not holding you responsible for the steps that I talked about specifically to them. But I do want you understanding that we're focusing on making factor 10. Now, yeah. Yes. Yep. Okay. Now, the initial activation of factor 10 only makes a very tiny amount of factor 10 active. Only the initial response only makes a very tiny amount of factor 10 active. Question back in the back? Okay. And factor 10 is what makes prothrombin into thrombin. So the initial response of the intrinsic and extrinsic pathways is to make a tiny amount of thrombin active. Now, thrombin activates a whole bunch of stuff. That includes factors that, in fact, activate factor 10. So thrombin activates those factors that activate factor 10. Factor 10 activates more thrombin, and this is that cycle I talked about of amplification. Notice I'm not saying the factors. You're not responsible for the factors. But this amplification happens because thrombin is activating factors that activate factor 10, and factor 10 activates thrombin, and we see that vicious cycle of amplification. Yes, ma'am? What are the factors? The factors are serine protease zymogens that get activated. So you saw that cascade. This goes to this, goes to this, goes to this. All those are serine proteases. And they're starting out inactive, and they get converted to active. Then that serine protease activates the next one in the cycle, activates the next one in the cycle. Make sense? That's what the factors are, for the most part. There's a couple factors that are not serine proteases, but for our purposes, you can consider them all to be serine proteases. Okay? Yes? So the pathways converge at factor 10, and everything after that are common. It's not one versus the other. So it actually comes back through the extrinsic, but you're not responsible for that. Okay? Yes? Can I briefly explain what the extrinsic and intrinsic pathways are? Okay. In simple terms, the extrinsic and intrinsic pathways are separate activating pathways that converge on factor 10. So the intrinsic pathway is the one that has many more steps in it. It involves serine protease after serine protease after serine protease doing that activation. What starts that? Well, okay, you're going to get into, see, I don't want to, why don't I talk to you separately about that? Because if I go through that in here, everybody's going to go, oh my God, we got to know all this stuff, right? So let's just save that, okay? Let's save that. Is that okay? And then we can talk about that later? Okay, all right, good. That's a very complicated story, okay, a very complicated story. All right, so are you with me now? We're at the amplification phase, a little bit of thrombin, is activating factors that activate factor 10, and now just the amplification over and over and over. The more that goes, the more thrombin we get. That amplification phase amplifies thrombin about the formation of thrombin about 1 millionfold. A ton of thrombin is made active. Now, the critical factor for that is that thrombin has got to be at the place where you want the clot to form. Because if thrombin is not at the place where you want the clot to form, it's not going to get amplified. I'm sorry, the prothrombin, back up, sorry, but I'll erase what I said. Prothrombin's got to be at the place where you want the clot to form, because that's where it's going to get converted to thrombin, right? So you got to have prothrombin there. And what's the signal about where this is going on? The signal is calcium. So prothrombin has got to be able to bind calcium so that it's held at the site of the wound. That's critical. Prothrombin binds calcium. How does prothrombin bind calcium? Remember the carboxylation of prothrombin? Adding the carboxyl group. Adding the carboxyl group allows it to bind calcium, and now prothrombin is there.
Isn't that after the soft clot has been made by everything?
No. That happens before the soft clot is made, because you've got to start making fibrin to make the soft clot. And to make fibrin, you've got to have thrombin. Can I repeat how it binds? Yeah, let me see if I can just pull up a figure instead of just talking words here. How about that? I think I've got that in here. Let's see. Oh, I don't have it on there. Hold on, let me think. Oh, I've got it in here. Hold on, I've got it in here. Yeah, it's easier if I show you this instead of try to do it in words. We're not making the soft clot yet. No. The carboxylation happens before the soft clot.
On the notes, doesn't it have it after?
That isn't necessarily sequential, but it happens before the soft clot. Yeah, definitely happens before the soft clot. All right, so let me show you what we're talking about here. Okay. Enzyme. I think it's this one. That's fine, okay. Yep, okay. Now, spots. And so we were, the original question, what was the original question? I forget what the original question was. Oh, how calcium binds. Okay, so that was actually right here. All right. So calcium is binding to prothrombin. Okay, right here. So what's happened is the side chain of glutamate of prothrombin has been carboxylated. Those two of them bind to calcium. Okay. That gets put on because of a reaction that requires vitamin K. That's why vitamin K stimulates blood clotting. Because now this guy is held at the site of the wound, and when it's held at the site of the wound, prothrombin gets activated to thrombin. The reason that's before the soft clot is thrombin. See, we've already held it at the site of the wound. Thrombin has got to convert fibrinogen to fibrin, and that's when the soft clot forms. So I talked about it afterwards because I didn't want to just throw it all out there in the lecture. I talked about vitamin K. In fact, if you look in the lecture, you'll see vitamin K pops up later in the lecture. We got it here somewhere. Yeah, there's vitamin K. Okay. I didn't talk about this until I got to vitamin K later, right? So, here's old Queenie. Okay. But vitamin K is necessary to hold that, put that carboxyl on there to hold prothrombin at the side of the wound.
So vitamin K is what? Yeah.
Is what?
Vitamin K is what carboxylates the prothrombin?
An enzyme carboxylate, but vitamin K is used by that enzyme. It's a cofactor. Yep. Okay? Yes, back in the back. That's correct. Yep. Because remember, calcium is released in the cellular response. Calcium is there. So here's a flag. Here's where this damage is, and prothrombin says, okay, I'm stuck right here. And now all this cascading is going on. Says, oh, here's some prothrombin. Convert it to thrombin. Thrombin converts fibrinogen to fibrin, and bang, we've got a clot. Right? The clot that forms is a soft clot. Right? Why do we have... I don't know why that's there. Okay. What's that? It's time for a metabolic melody. Maybe we can finish tonight with a metabolic melody, okay? All right. So 1 million-fold soft clot. Soft clot's got to be converted to a hard clot. That's the action of the enzyme transglutaminase. Oh, yes, question. Yes. Yep. The prothrombin's all at the side of the wound. It's all sitting there. So prothrombin's been sitting there just waiting for all this to happen. Prothrombin is floating in your bloodstream everywhere right now. So this is the really cool thing. It's a good question. This is the really cool thing about blood clotting. All these things are floating in your bloodstream all the time. That's why people develop a clot and it kills them, because the factors are already there. You don't want clots forming unless you have damage, right? And you've got to have all these things floating in your bloodstream all the time so that you're ready to attack, I shouldn't say attack, but to make a clot as quickly as possible. So they're not just being made when this happens, right? Prothrombin is just floating there all the time. You've got plenty of prothrombin floating around right now. Okay, how does prothrombin know to be there? Prothrombin is binding to calcium, and calcium is there because of the molecular response, right? So the molecular response, that calcium is critical for all these other things that happen, right? So that release of calcium says, hey, here's where the problem is, right here. Yeah. It does. Yeah, absolutely. Prothrombin's floating through the bloodstream, right? Some of it's floating out this hole in your vessel, right? You're bleeding some prothrombin that's going out, but some of it is sticking right there at the side of the wound. Yeah. I mean, this is amazing. You think about this happening. This is happening. You're forming a clot in a minute or two, okay, to stop you from bleeding to death, and that clot doesn't form any other time when your body is sitting here making all this stuff. It's just incredible that it happens. It's incredible that it's so rare that people develop clots that cause strokes and things like that. It's a wonder it doesn't happen more often. Yeah, question back here. Say it again. You're asking about warfarin, so let me come to that in a minute. Yeah, the warfarin is the one that inhibits the recycling of vitamin K. What did you say? Okay, so vitamin K gets made into vitamin K epoxide in the process of carboxylation. All right. So let me just finish this, and I'll talk about that. Okay. Okay. All right. So we have the transglutaminase. Transglutaminase is catalyzing formation of a covalent bond. That's why the clot hardens. What's holding that soft clot together are just hydrogen bonds, ionic interactions, and so forth. There's no covalent bonds formed until the transglutaminase comes there. This polymer, this fibrin polymer, is just all these fibrin subunits just coming together and just holding on to each other. That's all it is. That gets hardened by this transglutaminase, which now literally staples them together. It staples them together with a covalent bond. The covalent bond is between a glutamine and a lysine. That's why it's called a transglutaminase. And was there a question here? In fibrin, that's right. The glutamine and lysine are in fibrin. Okay. Make sense? Yes? Transglutaminase float. No, all these things are floating around. Transglutaminase is known as factor 13. What's that? Yep. So, it's a good point. Transglutaminase is activated by thrombin. It's one of the things that's activated by thrombin. Okay, still a lot of stuff, isn't it? I was hoping to make this simpler. You have my sympathies with this, guys. There's a lot of stuff here, especially if you're getting it the first time. Now, I want to come back to his point about the vitamin K in a second. Any other questions about this before I talk about the vitamin K? Yep. So transglutaminase forms covalent bonds that staple together the individual fibrin units, and that makes a hard clot. Okay? With me? Yes? Okay, well, good. Okay, I'm happy to hear that. I'm actually going to do this weekly. So I appreciate the feedback, and I will do this weekly. Okay? And that makes it, we're not going to have, tonight's going to be a big one. I hate to tell you. So tonight's going to be a big one. But when we do it weekly, it won't be quite so involved. Okay? And, yeah, this is great. Good, good. Yeah? Sure. So the side chain of lysine, which is that amine, okay, interacts with the side chain of glutamine, which has a carboxamide. You don't need to know that, but that's what it has. Okay, these two get joined together, and you split out an ammonia, and that's the transglutaminase reaction. So that's the covalent bond that's formed between the side chain of glutamine and the side chain of lysine. And this is happening in fibrin. That's what's making it be hard. Yep. And it's literally hard because it doesn't pull apart. Covalent bonds are strong. And you got thousands of them. You got thousands of these units, and every one of them's got probably multiple ones of these. So you've got a lot of staples in there, really. Within the fibrin polymer. That's right. Yep. Good. You guys getting it? Good, good. I'm happy. I'm so happy. I've been worried about this blood clotting thing for a while, so I hope that's helping. Okay? Good. Okay. The last two things I'll talk about on clotting, and then I'll move to the next subject. The next subject shouldn't take quite as long. Okay? The next thing I'll talk about is the carboxylation. The carboxylation of prothrombin. As I think I mentioned in the lecture, there are other proteins in the clotting scheme that also get carboxylated. I didn't talk about them. I'm not going to talk about them. But you can imagine that carboxylation is important for what reason? Binding calcium, holding all these things to the site of the wound. So carboxylation is really critical. All of them involve the carboxylation of the side chain of glutamate. All of them allow these various factors to bind calcium and be at the site of the wound. That carboxylation reaction involves an enzyme that uses vitamin K. Okay? Enzyme that uses vitamin K. Vitamin K is converted in that reaction to vitamin K epoxide, which is what you were talking about. And that vitamin K epoxide has got to be converted back to vitamin K. Okay? And you're going to ask me the name of the enzyme, and I can't remember the name of the enzyme. Yeah, you probably should know, but I don't remember it myself. So, but the point is that warfarin is inhibiting that recycling. Warfarin inhibits the conversion of the epoxide back to vitamin K. So if you don't convert it back, all your vitamin K gets converted to the epoxide, and then you get no more carboxylation. If you can't carboxylate, these things aren't going to be held at the site of the wound. You're not going to have clotting. So that's why you're very careful how much warfarin you give a person. You give them too much, they're going to bleed to death because they're not going to have any clotting at all. With me? Yes? Okay, so vitamin K is used by the enzyme that does the carboxylation of glutamate. Okay? Vitamin K is used by the enzyme to put the carboxyl group on the glutamate. Okay? In the process, vitamin K gets converted into vitamin K epoxide, and that has to be recycled. That's where warfarin comes in. Okay. The very last thing I talked about in blood clotting was how we break down clots. Clots are broken down by an enzyme called plasmin. Plasmin. I showed a scheme in class. It was a little complicated. I'm going to make it simple here. Plasmin is activated from plasminogen. Plasminogen is the zymogen form, and plasmin is another serine protease. These are all serine proteases in this blood clotting scheme. Plasmin is a serine protease, and what it does is it eats the clot. It eats fibrin. It's eating fibrin. Okay? This is the healing of the clot, right? All serine proteases breaking peptide bonds. So they're breaking peptide bonds that's eating that are part of the fibrin. Oh, that's a good question. Are the covalent bonds an issue? Those get broken down separately. They would be, but since everything is chewed up around it, then they don't factor into the equation. Yep. So you can imagine when you start chewing something off the protease, you get all these little pieces of peptides. And so these covalently linked ones are just little parts of the little pieces that are just floating around. All right, so plasmin is made from plasminogen. What converts plasminogen into plasmin? It's something called TPA, tissue plasminogen activator. That's the molecule I said. It's not a molecule, it's an enzyme. That's the enzyme that was the first genetically engineered product put into human beings. It's used to break down clots that are problematic in places where they shouldn't be. So like in a heart, where there's a clot, they want to get it out, they want to get it out quickly, or in the brain somewhere. They use TPA because TPA will activate plasminogen into plasmin and break that clot down. It's used very carefully because, as you can imagine, you don't want to just randomly get that going. Yes? TPA, all these things are normally circulating in the bloodstream, every single one of them. Yeah. How do you control all this chaos, right? It's scary. What's the difference between warfarin and TPA? Warfarin is a man-made molecule, so that's not part of the blood. Yeah, warfarin is a drug. Warfarin is a man-made molecule. It's the only man-made molecule I've talked about here. But warfarin is a man-made molecule that's inhibiting the formation of the carboxyl group on there. So it's an inhibitor of the clotting process, essentially. So TPA is the breaking down after a clot is made. One is preventing the clot. The other one is breaking down a clot after it's made. Make sense? Good question. Yes? Okay. Where do you get heparin from? Heparin's also floating around in your bloodstream. Yeah. It's scary, isn't it? Now, heparin is an anti-clotting factor because it's full of all those negative charges. And what do all those negative charges do? They grab calcium and yank it out of the process. So it's working counter to the clotting process. And one of the things that's happening with heparin, which you're not responsible for, but I'll tell you because I mentioned it briefly in class, is that heparin is inactivated early in the process by the von Willebrand factor. Right? Now you think of the orchestration that's got to happen in this. You think it's hard to learn. Think how hard it was to evolve, guys. This was a tough one. And if it doesn't work right, you're not around for very long, right? I think it's one of the most remarkable processes that happens in the human body. It's incredible. And we're still learning how the processes happen so that one doesn't step over the other and all of a sudden cause a problem. You don't want the clotting to happen when you don't want it to, and you don't want the unclotting to happen when you don't want it to, right? Both are problematic. You've got to have it timed just right. Yeah. How is TPA activated? I didn't tell you in class, but if I recall correctly, TPA is actually activated by thrombin. You're not responsible for that. Okay? Thrombin activates a lot of things. So thrombin, the thing that's making the clot, is also ultimately activating the thing that helps to break down the clot. Aren't you glad I simplified it? If you asked me to sit down and write down the clotting process, guys, I couldn't do it. And that's why I decided I wanted to make it simpler. I hope this is simpler for you. That's important. Yes? Okay, so the coordination of the breaking down of the clot has to happen, obviously appropriate for the healing process. And yes, it does. I have no idea, to be honest with you, how that happens. I did mention in class an enzyme that was important for that healing process. It had nothing to do with clotting. It was actually activated also by thrombin. Anybody remember what the enzyme was? Collagenase. And why was collagenase important? Where did collagen come into this picture? With the cut. You exposed the collagen. That was what initiated the process. So you got exposed collagen. When you have the healing process going on, you don't want that exposed collagen because what's going to happen? More clotting. It's going to clot again, right? So you got to knock that back out of the process. Collagenase, yeah. It breaks down collagen. Yeah. So collagen is exposed during the wounding process. Are we done with blood clotting? Okay. You have a question? Okay. That's fine. That's fine.
So would it be just safe to put that collagenase is like the very last step? It's like we're taking that.
I don't want to say there's an order to it, no. But collagenase is necessary for the healing process, definitely. Yeah. And it would be late in the process, obviously. Yeah. Yes?
What happens after the heart clot?
After the heart clot?
Or after it bursts?
Oh, the hard clot. I think you said the heart clot. The heart clot, you have a heart attack is what happens after the heart clot. It's not a pretty picture at that point. What happens after the heart clot? Well, the healing process is going on. So you've stopped the blood flow at that point. That hard clot forms within, a couple of minutes of the cut. So the healing process, of course, depending on the severity of the cut, can take anywhere from a couple of days to a couple of weeks to happen. And so that healing process is going to happen during that whole sequence. The clotting is happening much faster than the healing itself will actually occur.
And then once the heart clot is formed, the calcium and the signals go down.
Right. So when the heart clot forms, you're covering up all the area of the wound. It's not exposed, so it's not bringing all these other factors in. The clotting process is going to stop because it's got no signal. It's got nothing else that's going on, and everything is still floating around the bloodstream. And you hope to hell it doesn't go back there and cause a problem, because if it does, if it blocks, for example, that arterial flow, then you got a problem, right? So you want to have it just enough to stop that flow without having everything else going on. That's right. So the clot is covering that stuff up. Okay? Yeah? How do calciostatin and say them again. I can't remember the names myself. Osteocalcin and periostin, right? And how do they? All right, I'm sparing you that. All right. Now, those actually play roles in the bone-making process. All right? So the bone-making process, bones are really critical because bones are your stores of calcium. You think of them as your support systems, which they are, but your bones are giant reservoirs of calcium. And you're making and breaking down bones as the body's need for calcium is either high or low. If you're getting plenty of calcium in your diet, your body is going to start putting it into actually what it's going to do, if you start getting plenty of calcium in your body, the body's going to start excreting it, right? And if you don't have enough, then your bone's going to break down and release calcium so the body can use it. It has to do with calcium storage there. So I'm not going to talk about that. Okay. All right. Was that more? You guys like blood clotting? Or you're terrified of blood clotting? You think, God, I got to know everything about this or he's going to kill us on the exam, right? All right. I promise I will be fair in terms of hitting high points on exam. I'm not going to go into dreadful detail on blood clotting. I'm not going to ask you to draw the process. All right. So I will use high points of what I've talked about in blood clotting that I will expect that you will know. Okay? You're not going to need to know in any more depth than what I've had here. Okay? Okay. All right. Carbohydrate structure. Review of organic chemistry, right? So, hexoses, pentoses, tetroses, heptoses, octoses. Everybody knows what those are, right? 6, 5, 4, 7, 8 carbons. Os meaning sugar. Right? Monosaccharide also means sugar. One sugar unit. Glucose is a monosaccharide. Ribose is a monosaccharide. galactose is a monosaccharide. Fructose is a monosaccharide. And those are the 4 monosaccharide structures you should know, both in the linear and in the circular form. Oh, I misspelled glucose, didn't I? Gluxo.
So you say we should know them, prepare to draw them?
Yep, yep. So I expect that you should be able to draw those. Yep, because I'm a mean guy. Okay? All right, so those are also aldose, 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 ketose. An aldose has an aldehyde group at the end. A ketose has a ketone. Yes? What? Say that again? So you need to draw linear and circular of all those. Yeah. Okay? Okay, so you only need to know the furanose for fructose and ribose, and you know the pyranose for the other 2. Okay? Aldose, ketose. Stereoisomers. Stereoisomers occur whenever you have a carbon bound to 4 different things. Sugars are full of stereoisomers. If we have 2 sugars that are mirror images of each other, they are known as enantiomers. L-glucose and D-glucose are enantiomers. If we have 2 sugars that differ only in the configuration of 1 carbon, they are known as epimers. If they differ in the structure of more than 1 carbon and they're not mirror images, they're known as diastereomers. And if they differ only in the configuration of their anomeric carbon, they're known as anomers. Alpha D-glucose and beta D-glucose are anomers. You can always tell anomers because they have the same last name. It's like cousins marrying or something, right? I don't know what that means, but it sounds kind of funny. All right, cyclization. You should note the straight chain form is known as the Fischer form. The circular form is known as the Hayworth form. A furanose is something that has a 5-membered ring. And a 5-membered ring doesn't mean 5 carbons. It means a 5-membered ring. A pyranose is something that has a 6-membered ring. And that doesn't mean 6 carbons either. Okay, I talked about glycosides. A glycoside, anybody want to give me the definition? How do I make a glycoside from glucose? What would I do to it? Which hydroxyl group would I alter? Anomeric. Very good. Thank you. Thank you. It's a little getting late, isn't it? The anomeric carbon. If I alter the anomeric hydroxyl group in any way, I make a glycoside. I mentioned a couple of artificial sweeteners. I talked about sucralose. And what was the other one I talked about? I don't think I talked about NutraSweet. What was the other one I talked about? Doesn't matter, but it's in your notes somewhere. It's in the highlights, I know that. Okay, disaccharides. The only one I expect you to know the structure of is sucrose. Sucrose has glucose in the alpha configuration, fructose in the beta configuration. It's what I called a diglycoside because you have the hydroxyl of the glycoside of the anomeric carbon of glucose linked to the hydroxyl of the anomeric carbon of fructose. They're joined together through that oxygen. Both are altered, so it's a diglycoside. Sucrose is a non-reducing sugar. It has no free anomeric carbon. All the others I've talked about are reducing sugars. They have a free anomeric carbon. The free anomeric carbon is important because that allows the sugar to go to the straight chain form and form either the aldehyde or the ketone. And that's how it gets oxidized. Two other disaccharides I talked about were lactose and maltose. This isn't the order I talked about them in. I talked later about oligosaccharides, but I put it in here because I thought logically it made sense to put it in here. Oligosaccharides have small numbers of sugars joined together. And we find oligosaccharides in glycoproteins. What is a glycoprotein? A glycoprotein is a protein that's attached to an oligosaccharide. And glycoproteins are important for identity purposes. We find them on the surface of cells frequently. And they're like a license plate tag that says, I'm here. I got to check the camera and make sure I've still got enough juice in there. Otherwise, we're going to run out of juice, and the people in TV land are going to be very unhappy with me. And I don't like it when TV people in TV land get unhappy with me. 54 minutes. Okay, we're fine. Okay. So that's what a glycoprotein is. We see glycoproteins on the surface of blood cells. They give blood cells their type. We see glycoproteins on the surface of cells like tissues. So, for example, kidney cells have glycoproteins on their cell surface. Your body says, this is mine. It doesn't cause a problem. I take the kidney out of me and I give it to you. If my pattern of the oligosaccharide on my kidney doesn't match that of yours, your immune system says, enemy, and attacks. And that's what causes tissue rejection. So you've got to have the right types. That's why relatives usually are the best people to get transplants from, because they have the same similar pattern of oligosaccharides on those critical tissues. Okay. I talked about polysaccharides. Polysaccharides, of course, polymers of the saccharides. Cellulose, polymer of glucose. Beta linkages. Beta-1,4 linkages. We can't break those down. We don't have the enzyme called cellulase that's necessary to break down cellulose. Ruminant animals have bacteria that contain cellulase in their rumen. That's how they eat grass and get energy. That's how they get gas. Glycogen, polymer of glucose in us. It's our storage form. It's the primary storage form of glucose in human beings, in all animals. Alpha-1,4 linkages, alpha-1,6 branches. Those branches occur fairly readily in us. About 1 every 10 glucoses, we get a 1,6 branch. The more branches, the more ends we have. Plants have a branched form that's related to ours. It's called amylopectin. They also have alpha-1,4 linkages and alpha-1,6 branches, but the branches there are about every 30 to 50 glucoses. Not nearly as branched as our glycogen is. Keeping up? Am I going too fast? Got it all down? Amylose, polymer of glucose, only alpha-1,4 linkages. It's also found in plants. And amylose and amylopectin are what make up starch. Yeah? Sure. Sure. So let me see if I can pull it up on my thing over here. Hold on. No, amylose only has alpha-1,4 linkages. Yep. All right. Keynotes. Okay, let's go to carbohydrates. That would probably be in the second carbohydrate lecture, which would be right here. Okay. And where am I at here? Glycosaminoglycans, disaccharides, reducing versus non-reducing, polysaccharides. Okay. So, all right, there's amylose right there. Alpha-1,4 linkages only, no branches. Okay? Glucose, glucose, glucose. Alpha-1,4. Amylose, another view of the same thing. Amylopectin. Amylopectin has alpha-1,4 linkages that go across the bottom, and the branches are up here. So we see that's an alpha-1,6. There's 1, and there's this carbon number 6. Okay? And notice every 30 to 50 residues. Okay? So amylopectin is not nearly as branched as is glycogen. The numbers refer to 1 and 6. Okay, good question. So an alpha-1,4 bond, here is carbon number 1. It's in the alpha configuration because it's down. Here's carbon number 4. Carbon number 4 is always down in glucose. So these are both pointed down, and that's an alpha-1,4 linkage. Okay? alpha 1, 6. My hand doesn't go up that high. There's the alpha right there, all right? In the down position. 6, of course, is in the up position, and we see alpha 1, 6. Make sense? Good. That's fine. No, I'm happy to clarify it, yeah. Yeah. The difference between amylose and glycogen is that amylose has no branches. No branches at all. So amylose is simply an alpha 1,4 polymer, pure alpha 1,4, no branches at all. What's that? Beta 1,6? No. I talked about a beta with respect to sucrose. So sucrose has the hydroxyl and a beta configuration of fructose and then the alpha configuration of glucose together. But I don't have any beta 1,6s now. So beta 1,4 for cellulose. So if I go to cellulose, let's see, I forget where it's at. Actually, here's my thing. So there's alpha 1,4s, you see. There's alpha 1,6s, right? Okay. There's glycogen, so we see that's every 7 to 11 instead of every 30 to 50, right? Glucose, alpha 1,4, alpha 1,6. Okay. Chitin, did I skip? Yeah, I did skip the, hold on, get back to the there's cellulose. Okay. So cellulose has a beta 1,4. See it going up off of the 1? That's the anomeric carbon, so that's the beta configuration. Beta, 1, and then there's 4 again. Okay? All right. Let's see. Let's get back to what I had here. What's that? I think you should know the names of the linkages. I'm not going to ask you to draw them, but I think you should know the names of the linkages. Yeah, I think that you should know that cellulose is a beta 1,4, and that's what a cellulase works on. Yeah, sure. Yeah. Okay. There's the polysaccharide. Chitin was the last one I didn't mention. Chitin is the one that makes the exoskeleton of insects. It's a polymer of N-acetylglucosamine. And I don't expect you're going to know the linkage to that one. I think it's alpha 1, alpha 1,3 or alpha 1,4. I can't remember, but I don't expect you're going to know that one. Okay. Generally, if I don't remember it, I don't think it's fair to ask you to. And I've been teaching it for how many years? So, all right. Glycosaminoglycans. Now, that sounds a lot like glycoproteins, for example. It's not. So a glycosaminoglycan, the name tells you what it does. Glyco, sugar, amino, that has an amine group on it, glycan, polymer of sugar. Glycosaminoglycan. It's a long polymer that contains an amine sugar. Glycosaminoglycans will always contain negative charges. Negative charges. They're polyanionic. They contain thousands of negative charges. Heparin is a glycosaminoglycan. Yes? Okay, where are the negative charges? I'll have to show you the structure. Let me pull up a structure here for you. So the structure, come on, Keynote. Okay. Chitin, glycolipids. Did I skip over the glycosaminoglycans? There we go, glycosaminoglycans. Okay. Okay. There is the glycosaminoglycan component of hyaluronan. You can see there's the amine sugar right there. There's the negative charge part of it right there. So they will usually involve, this is glucuronic acid. I didn't tell you that, so I don't expect you're going to know that, but they always will have something that's negatively charged, and they will have an amine. So that's what a glycosaminoglycan is going to have. Okay? When we look at heparin, and I'll show you, actually I will show you heparin because I showed you that one in the class. There's chondroitin sulfate. You can see the glucuronic acid, and you can also see the amine sugar over here. Okay? There's heparin. Look at heparin. Heparin's got sulfate, very negatively charged. It's got sulfate, very negatively charged. It's got sulfate, very negatively charged. It's the most polyanionic substance known. More negative charges per density than any other molecule. And this guy right here, as I said, look at that. Just a perfect binding place for calcium. That's how it's acting as an anticoagulant. I'm not sure the first part you said about the heparin. No. These are modified sugars. That's a sugar right there. So this is the sulfate on the sugar, right? So the difference here, you got a carboxyl on the sugar. Down here, you got a sulfate on the sugar. You not only got one, but you got 1, 2, 3 per every 2 units. Okay? So these are not amino acids. These are glycose. So let me make clear. A glycose aminoglycan has no protein. They're all sugars. If I make a proteoglycan or a peptidoglycan, and I use those two terms interchangeably, I would make that by attaching this to a protein. I'm going to attach a glycosaminoglycan to a protein. That's how I make a proteoglycan or a peptidoglycan. Okay? Yes, back in the back. Glycose meaning sugar. Okay? So these are sugars, right? Amino referring to this amine right here. And glycan refers to the fact that it's a polymer. Okay? So the first part and the last part are kind of redundant. The first part doesn't tell you it's a polymer, but the last part says it is a polymer of a sugar. Yeah, okay? A proteoglycan is a protein attached to one of these sugar polymers. Right? A peptidoglycan, same thing. They're just calling it a peptide instead of calling it a protein. A peptidoglycan attached to this one of these sugar polymers. It does. So these are polymers of the polyanionic things. Yes. Yeah. Not all glycans are polyanionic. That's right. They're not. Okay? You don't need to know that, but that's true. So there are glycans, for example, that are in your teeth that bacteria make, right? And they're not these guys. They're long polymers of sugars. They use them for energy storage. They cause plaque. The plaque on your teeth, that comes from glycans. Yeah. Okay? Clear as mud? We got to sing a song at the end of this, I think, guys. Okay. Let's see. Where was I? All right. Proteoglycans, peptidoglycans. Hyaluronan was the one I kind of messed up in class. I called hyaluronic acid hyaluronan. And hyaluronan is the protein attached to hyaluronic. It's what you get when you attach a protein to hyaluronic acid. That's what hyaluronan is. Okay. Hyaluronic acid is the glycosaminoglycan. Hyaluronan is the peptidoglycan. Yes, ma'am. We can do the serine protease song. We can do 2 serine proteases. So I have 2 serine proteases songs if you would like, okay? All right. All right. Let's get through this, guys. All right. Let's see. Briefly, I talked about lectins. Lectins are proteins that bind to specific carbohydrate residues, typically polymers. All right? Lectins are important for attaching to like cell surface components. Bacteria use it to hold on to your gut so they don't get squirted out with your poop, right? E. coli stays in your gut because they have lectins that are holding on to the carbohydrate residues of your intestinal wall, right? Residue is just chemical, right? So A residue is just, could be a sugar, could be an amino acid, could be anything. It's whatever is just hanging out there. Yeah, that's what a residue is. It's what you have left in your sink after you don't clean it up. Same thing. All right. All right. Okay. Let's see. I talked about flu and Tamiflu. Flu uses what's called a hemagglutinin to attach to a cell. That's where the lectin component comes in. It's called a hemagglutinin to attach to a cell. It injects its RNA into the cell, and flu virus is an RNA virus. You don't need to know that. All right. The RNA goes in, gets translated and replicated, and all the flu virus particles are made. And to get back out of the cell, they've got to use an enzyme called a neuraminidase. Neuraminidase. The neuraminidase is necessary for the flu virus to exit the cell. Tamiflu inhibits the neuraminidase. So, flu virus, can't get out of the cell. It ends up agglomerating inside the cell and is not active, and therefore you kill the virus. So that's what Tamiflu does. Okay. All right, I talked very briefly about glycolipids. Name tells you what it does. Glyco meaning sugar, lipid meaning lipid. Sugar attached to lipid is a glycolipid. Yes, ma'am? Yes. So the neuraminidase has to act for this virus to get out of the cell. What it's doing is it's clipping, I'll tell you, you don't need to know this, but it's clipping some sugar residues. So it has to clip those sugar residues before it can get out of the cell. If it can't clip those, it doesn't make it out of the cell. And so by inhibiting that, you keep it in the cell. Okay? All right. Glycolipids. We'll talk more about those next term. I briefly mentioned glycerophospholipids and sphingolipids. All right? Just categories of glycolipids. All right, G-protein coupled receptors. We're getting there, guys. Okay. G-protein coupled. I'm looking at that clock and I'm thinking, oh man, because that clock is an hour ahead. They haven't set the clock back, so it's only 8:30 instead of 9:30, but still. Okay. G-protein coupled receptors. All right, we talked about this just most recently, so I'm going to go over this relatively rapidly. Okay. It's the receptors that are found, in the case of G-protein coupled receptors, in the membrane of a cell. They bind to a hormone that comes outside. The hormone is the first messenger. G-protein coupled receptors stimulate production of second messengers. Second messengers are small molecules made in response to the binding of a hormone. Okay. Couple hormones that act in the same way, epinephrine and glucagon. Both of them ultimately stimulate production of glucose. I talked about how they're activated by the binding of the hormone. I talked about how that stimulates the activation of a G protein. And the G protein, you recall, has an alpha subunit that's inactive when it's bound to GDP. The binding of the hormone stimulates it to let go of GDP, take up GTP. That causes it to let go of the beta and the gamma subunits. The alpha subunit goes over to an enzyme known as adenylate cyclase. So the night's getting long. Adenylate cyclase catalyzes the formation of cyclic AMP. Cyclic AMP is the second messenger in a G-protein coupled receptor system. Yep. Okay. Adenylate cyclase makes cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A has a catalytic subunit, regulatory subunit. When the catalytic subunit is bound to the regulatory subunit, no activity. Cyclic AMP binds, regulatory subunit lets go of the catalytic subunit now active. Catalytic subunit is a protein kinase. Protein kinase tells you what it does. Puts phosphates onto proteins. Protein kinase A puts phosphates onto two proteins I talked about in class. Phosphorylase kinase glycogen synthase. It inactivates glycogen synthase. It activates phosphorylase kinase. Phosphorylase kinase phosphorylates glycogen phosphorylase. Glycogen phosphorylase breaks down glycogen. Bang, you got glucose. You guys getting tired? Me too. Yes? So protein kinase A puts phosphate onto two proteins. One protein is phosphorylase kinase. The other protein is glycogen synthase, the thing that makes glycogen. That has the effect of turning off glycogen synthase. Okay? If you're going to break down glycogen, you don't want to be making glycogen at the same time. Right? Phosphorylase kinase puts a phosphate onto glycogen phosphorylase. Glycogen phosphorylase breaks down glycogen. You get glucose ultimately out of that. Okay? Inactivate it. Several ways of inactivating that system, and all of them are important. Arrestin, a protein that comes along and recognizes what? What does arrestin recognize? You may remember? Phosphate on the G protein coupled receptor, right? That got on there by a kinase that put a phosphate on there. That knocks out the receptor from functioning. Alpha subunit has a GTPase activity. It breaks GTP down to GDP slowly. That causes the G protein, the alpha subunit, to become inactive. No longer stimulates adenylate cyclase. That takes adenylate cyclase out of action. Phosphodiesterase. The cell is full of phosphodiesterase. It's always breaking down cyclic AMP. All right? So the cell makes a burst of cyclic AMP, and the phosphodiesterase catches up, and it starts breaking it down. So we see cyclic AMP go like this, up, and then finally it goes down. Because when we stop making it, this phosphodiesterase catches up. Okay? Phosphodiesterase inhibited by caffeine. So if you inhibit phosphodiesterase, cyclic AMP concentrations go up. Cyclic AMP concentrations go up. Protein kinase goes up. All these other things go up. More glucose. More glucose, more buzz. Caffeine gives a buzz. Yes? Cyclic AMP is broken down into AMP. So it's the cyclic bond that's broken. It forms AMP in the process. Okay? Arrestin, I won't say is slow, no. So arrestin's, the thing that's slow is the GTPase activity of the alpha subunit. Okay? Phosphodiesterase is sort of constant, right? So if it's constant and the production of cyclic AMP is constant, then the level of cyclic AMP is going to be pretty constant as well, right? But you get a burst that comes from a hormone binding. Cyclic AMP goes up, and then when the signal is turned off, you see the adenylate cyclase stops making it, so the phosphodiesterase starts catching up, and it goes back down. So you see cyclic AMP go up, and then it comes back down. You go up, and then it sort of comes back down. So arrestin recognizes the phosphate on the G protein coupled receptor. That's right. So there's a kinase that puts a phosphate onto that receptor. Okay? Yeah. This is it's a lot. Well, that's good, but we're getting through it. Okay. Receptor tyrosine kinases, RTKs. These guys are also membrane receptors. They have to be dimers to function. And when they're dimers, they autophosphorylate, meaning that they put phosphates on the tyrosines of the cytoplasmic tails of each of them. That activates them. This process is called autophosphorylation. For most of the RTKs, they dimerize only when they bind to the appropriate hormone. That appropriate hormone, in the case of epidermal growth factor receptor, is epidermal growth factor. In the case of insulin receptor, it's insulin. Okay? Insulin, however, insulin receptor is already a dimer. So the binding of the insulin, all it does is it causes the autophosphorylation. It doesn't cause the dimerization. But in each case, the binding of the hormone is responsible for the autophosphorylation, And the autophosphorylation is necessary to put the phosphates on those tyrosines, which become targets for the signaling complex. The signaling complex ultimately is what causes the cell to do what it does. Yes? What's autophosphorylation? I'm not going to pull up a figure and then I'll try to describe it to you. I've got a membrane. Embedded in the membrane, I've got, let's say, over here, I've got the half of the epidermal growth factor receptor. Outside part up here, inside part down here. Outside the cell, inside the cell. Okay? I've got another one over here. This guy says, oh, look, I got an epidermal growth factor. It's got a ball in this hand here. It goes floating around, and these guys bump into each other and says, oh, this guy grabs the ball too. So they're now holding onto the ball. The two tails on the inside are close to each other, and they each have the ability to phosphorylate the other one. They put a phosphate onto the tyrosines of the other one. That's what the autophosphorylation is. And so because they're brought into a close proximity by the dimerization process, now they can do that. If they're floating around, they don't see each other. But when they bind, oh, let's phosphorylate, right? Does that help? Audio visuals are really important. Okay. Okay. So some of the examples of receptor tyrosine kinases. Two I talked about. Insulin receptor. Insulin receptor, I said, was always a dimer. It doesn't require insulin to dimerize. But it does require insulin to autophosphorylate. All right? It goes through quite a few steps. I don't expect you're going to know the steps. You don't need to know the steps. But you do need to know the outcome. The outcome is it moves a protein called GLUT4, whatever you want to call it, to the cell surface. GLUT4 is a glucose transport. That's what GLUT stands for, glucose transport. And now when that sucker's on the surface of the cell, whenever it sees glucose, it brings it into the cell. Insulin has the effect of reducing blood glucose because by moving receptors to the cell surface, the cell starts taking in glucose out of the bloodstream. Blood glucose levels fall, cell glucose levels rise, but not for very long. All right? One thing that happens is it gets made into glycogen, which is no longer glucose, no longer poisonous. The other is glucose gets used in glycolysis, which also reduces cellular glucose concentrations. We'll talk about that actually starting next week. Everybody with me? Okay, bear with me, guys. You're patient. That's good. Okay, I'll talk about it later. I just briefly mentioned it in class, which is why I put it on here. It activates an enzyme called phosphoprotein phosphatase. Insulin binding activates this enzyme. The only reason I mention it, this enzyme takes phosphates off of everything that the kinases put phosphates onto. So insulin is doing the exact opposite of what the epinephrine and glucagon signaling was doing. Epinephrine and glucagon signaling was favoring the release of glucose. Insulin is favoring the uptake of glucose. Exactly the opposite. Epidermal growth factor, very important for cells' decision to divide. Decide or divide, yes. This one does require its hormone, EGF, to dimerize. That dimerization causes autophosphorylation, assembly of a complex, and all the signaling that happens from that. The difference here is that